Okay, we're going to cover Chapter 5 today on chromosome mapping. This chapter is pretty simple in terms of the concepts that you have to know, but it gets very complicated applying these. Students often find this chapter to be very confusing, and kind of the only way to get comfortable with the material in this chapter is to do lots of example problems. So this will be both in your activities and then also the mastering genetics homework. So the goals of this chapter: explain why linkage distorts gamete production in heterozygous individuals, calculate the map distance between genes on the same chromosome, establish the order of genes on a chromosome, and then use Punnett square to estimate genotype frequencies for linked genes. And the majority, actually the entirety of this chapter is going from the assumption from Mendel's work from Chapter 3 that if you have multiple genes, they're on different chromosomes. So again, this was the pea plants. You might have been interested in the color and the shape of the peas, the seeds that are produced. So green versus yellow seeds, and then rough, wrinkled versus smooth shape. And we kind of assumed that the genes that controlled those characters were on different chromosomes. And so this led to the idea of independent assortment. So when an individual that was heterozygote produces gametes, all four possible gamete combinations were produced. So you could pair the capital A with the capital B, but you could also pair the capital A with the little b, and then the remaining two other combinations. So now we're going to go to the situation where the genes are actually on the same chromosome. So gene A and gene B are both on chromosome 1, and again we're looking at a heterozygote individual. So there's only two combinations that are present. Capital A is linked to capital B, and that's because they're physically present on the same chromosome. There's a physical linkage between those two alleles. And in the homologous chromosome, you have the little a allele linked to the little b allele. And so let's consider first the situation where no recombination occurs. So we know that in prophase 1 recombination occurs, but let's pretend for a moment that it doesn't. So what sort of gametes would be produced by this individual? And there's actually only two types of gametes. So when these homologous chromosomes separated, you would keep the linkage, the original linkage between the alleles that was present on the chromosomes. So you'd only be creating two types of gametes, the capital A paired with the capital B, and then the little a paired with the little b. And so this is kind of the situation that motivates the material from this chapter. And so we're going to break this down into two sections. So the first section will cover looking at two genes at once, and then in part 2 we're going to look in situations where there's actually 3 genes all on the same chromosome, and the different types of experiments that you can do in those situations.